Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Our Dairy NRC series of Real Science webinars was very well received, and tonight we're talking about the chapter on protein and amino acids. Hi, I'm Scott Sorrell, one of your hosts tonight here at the Real Science Exchange. Tonight, we're welcoming Dr. Mark Hanigan. As the author of this section, we're looking forward to a very lively discussion with him and his guests. Dr. Hanigan, welcome to the Real Science Exchange.
Thank you, Scott.
Before we get started, can you tell us a little bit about your background, Mark, and how were you selected to be the author of the proteins and amino acids section?
I really have no idea on that. I mean, it's probably like, who do they think they can talk into this, massive amount of work, and it's too stupid to say no. So.
Yeah, very well. And you're currently at Virginia Tech. Tell us what you do there.
I'm a professor in dairy science. I think we're maybe the last separate department, but we're in the process of merging with animal science. So.
All right, very well. Mark, it's been over 20 years since the last NRC, now called NASM. And I'm sure this required a lot of time and effort from both you and the rest of the team to create the latest edition. Speaking of team, I understand that you've brought several of them with you. Would you take a few moments to introduce them?
The colleagues and people that did a lot of the work as well include, of course, all of the committee members, but Dr. Jeff Firkins from Ohio State has joined us today, and so has Dr. Hélène Lapierre from Ag Canada or Agri-Food in Canada. I forgot what the appropriate name is.
That's why they change it all the time.
Just when you get used to it, huh? And of course, everyone did a massive amount of work. Maybe Jeff and Hélène had a better idea of how much work this was going to be, but in typical fashion, I underestimated the amount of work by at least 3 or 4-fold, more than I typically underestimate. So.
Yeah, understandable. So welcome, Hélène and Jeff. I look forward to having a lively conversation with you. Hélène, I understand you're taking this virtual pub thing seriously. What are you drinking tonight?
Oh, I had to prepare something that was made from Quebec. So this is a mixture of fresh lemon juice, and a nut liquor, which is made in Quebec, and a little bit of maple syrup. So it's really refreshing, really dangerous in summertime because you just feel it's like lemonade, but a little bit of ice, and it's really, really good.
Sounds good.
I even brought the bottle. I have to show you the bottle because it's really nice.
Very nice.
Nice.
Yeah, excellent.
So really good.
Yeah. Jeff, welcome. It's always nice to have somebody from The Ohio State University. I'm a grad myself. Well, wait a minute. I haven't introduced Clay yet. Clay, he's back in the co-host chair. Welcome. And Clay, what are you drinking tonight?
I have a new apple cider here, courtesy of Stacy, our producer. An apple cider from Kansas.
Mark, to get us started, I'd like to dig into the approach and the process you employed to bring the protein and amino acid chapter together. What was that process? Where did you start?
Well, I'd have to start off and say that, this was an effort, at least on the writing and of the three of us, really. And so I was not the lead on this chapter, actually. I was the lead on the modeling chapter.
Okay.
And so I ended up sort of being the caretaker of the equations, I guess, when they were developed. And I did work on some, but it's certainly Hélène and Jeff had a huge part in this. And they did way more of the writing on that chapter than I did. I made a small contribution.
One of the things I'll just jump in and say, we started from the beginning developing a bunch of metadata to be used. So there was a lot of work. Some of that was by the NENP. I guess that's how you say it. Mark was a leader of that. I joined in. We collected a lot of data and sort of, you would say, cleaned it and refined it. And a lot of that was done with in coordination with the way Mark's system was set up so that ultimately could be used in this larger model. So there was a lot of background, a lot of players involved in that. And that really was a big advantage of this time compared with the last NRC.
Yeah, fortunately, Gail Bateman, who was actually a postdoc with Jimmy Clark, and he's passed away now, but he was at LSU at the time, or later he was. He captured all the data from the last NRC. Otherwise, I'm not sure Jeff would have had it, because he's the one that had it, and it was some 10 years later when we sort of drug that back up and worked on getting it all cleaned up and getting all the diets sorted out as much as we could. I mean, that's the problem with all this, is that with entry of data, you're always going to have errors, and that even that data that had been around a long time, we found a few errors in that late in the process as well. And of course, all the data entry that had to happen was, Jeff had a student that worked on it. I had a couple students work on it. We had a couple postdocs from NANP work on it. I mean, I entered data. I mean, everybody seemed to enter data. So it was a large effort to do that, to get started, really. And then once you have the data, then you can start asking, okay, well, what was wrong with the last set of equations, or the last model, or what parts needed to be fixed? And, I think, Hélène, we decided that pretty much all of them had to be fixed, that they all had, updates that needed to be done. So, but we didn't throw out the system. I mean, it's the same basic system we had before, with more amino acid stuff in it now.
Can you elaborate just a little bit on maybe 2 or 3 big sections that you identified first that you needed to work on first to overhaul?
Well, the whole thing is a bit of a pyramid scheme, right? I mean, you start off with ingredient information, and then you try to track that through the animals. So, we had to get the feed library updated first, and so Jeff worked a lot with Paul Kononoff on that, and got the feed library updated. And Hélène and I tried to do stuff, but we ended up repeating things quite a few times. It seems like that it was like the movie Groundhog Day, where we kept getting up and starting over again. But eventually we got there. But the feed library got sorted out. Then we identified that there was challenges both with the microbial equations and with the RUP equations, and so Jeff took the leads on those. He also took the lead on the digestibility. We needed to update the digestibility data for the RUP and for the microbes. The composition of the microbes needed to be updated. I think Hélène worked on that as well. Once it gets absorbed, then we needed to divvy it up into the various processes. So I worked on the milk protein equations, and Hélène worked on pretty much all the other equations for all the maintenance requirements. And I think, Jeff, probably the growth sort of got left to the end. It sort of trailed along later and kept raising its head again, at least for me anyway, on the model side. It was like a bad stepchild. It just wouldn't go away. So.
Well, I'll jump in for a second, Scott. One of the things that I think was really an advantage moving from the old system to the new one. The old one was really, factorial in nature on a protein basis, but not on amino acid basis. That it had sort of a slope response way to do that with lysine methionine. The new one, is much more, in my opinion, sort of structurally related to a true requirements system. So I think there was, by having virtue of all those data to be able to fill in the gaps, we were able to do that. So Hélène and Mark really, if in my opinion, that's what they really added that really beefed up this new one.
Can you talk a little bit about how you acquired the data? And I'm going to assume that you scoured the databases, you looked worldwide, and what's the process look like for coming up with the data, putting it in a form that you guys can use and then put into the models?
But, you guys can feel free to jump in, but I mean, we had, a couple hundred studies to begin with from the old NRC. And then there was several efforts by multiple people to identify additional data. So I think Jeff and his student went through and looked for all the flow data that had been published since that time and anything else that got missed. And then Lou Armentano and a couple people on the fat chapter did a search, for all the fat digestibility data. And then I had a student that looked for all the amino acid data. And then we left off the credits list, Roger Martineau, who's worked with Hélène for a lot of years, did another effort on his side. Plus, he did a tremendous amount of work on cleaning data and identifying problems. And so there's probably so many people that got involved, it's hard to really lay your hand on everybody.
Yeah.
But it starts with a literature search, and then entering the data, and then checking the data, trying to see if there's any entry errors. And also whether the studies, a few studies got thrown out because they just there's something wrong with this study. I don't know what it can be. But...
So how many studies were included for the protein amino acid model for the NRC?
Well, it depends on the part, but for the overall effort, we're looking at Hélène, I think it was 365 or 385 studies, something like that. There's over 1,000 treatment means. We pay attention to that more probably than the studies. But of course, there was 580, I think we used for ruminal outflow. And then there was some of those were, fat oriented, fat digestibility. They still had milk production, so we still used it for that, right? But they weren't protein studies. There was some fiber studies as well that we used. So I can't say directly how many of them were protein studies, but certainly more than 600 treatment.
Yeah, and maybe I would just add as well that through this 7-year process, we've been often teased that it was taking a lot of time to go through all that, but we really felt that we wanted to start from the very beginning to assess where we were going and have the right data. So it took a long time. It just seems like a database, but we put a long time just to fix the database. And what we realized is that sometimes we wanted to have a too quick start, but then you do everything, but you realize that there were a few things that you were missing in the database, a few errors, and then you have to restart everything over. So I think that it was really a critical element of the whole model to have a good straight database that we could trust, from A to Z. And it took a lot of time. And as Mark said, that was all of us, it was our time, the time of our students, of our postdoc that weren't supposed to put their time in that, but that we just pulled in checking. Nobody's listening to us now. But I think that this has to be recognized that, it took some time, but we really, we worked hard to make it happen. So.
You talked about missing data. I'm curious, as you're looking at the data sets and you start plugging things together, did you identify any huge gaps that we just don't have that information? And then I'll ask you, what were those gaps? And then maybe a follow-up question, what needs to be done for the next NASEM that we need to follow up on and to make sure that those data are acquired?
One of the gaps I feel is there really isn't very much reporting of actual amino acids themselves. We had to factor amino acids from different ingredients from a database, and so a lot of people aren't reporting those data anymore. We have to rely on doing that. We have to assume that amino acids have the same degradability in the rumen and digestibility in the small intestine of the RUP fraction as the protein itself. One of the limitations, we really would like to be able to say one amino acid is different than another, maybe depending on the feed. So I'm not sure that's going to be fixed in the near future, but it sure would be nice to go back to having actual amino acid flows and some of that kind of thing. That's one thing that comes to mind. So we made some nice efforts to improve it. I didn't do this, but Hélène, did is the way that you measure amino acids to account for how they're lost during the analysis process. That was very much improved. So there was a lot of real science gains in there. The techniques are there for people to use. So we just need to be able to find, some people to get funding to do some of those key studies.
So as far as the ingredient database, what parameters were new in this version versus the 01? I know starch was available now, and that wasn't available back in 01. what are some other parameters that are new?
Well, I guess starch is a big one, and so that definitely is so. One of the things I hope people can do in the future is do a better job of measuring the residual organic matter fraction. So some of that's laid out. We moved to a fatty acid basis, so there's more fatty acid data for digestibility of fat. I guess one of the big things is, the previous committee really did a valiant effort to try to get data for digestibility of the RUP fraction. There weren't very many sources of data out there then. And so we really filled in so in the previous one, they just to signal users that we don't really know, they had these digestibilities in increments of 5 units. And so that's much more robust than it was since then. Mark had a hand in some of that too, probably. But so anyway, the database itself is much stronger, but we still have a ways to go. In some feeds, there weren't very many observations, so some of that could be filled in.
I'm curious if the previous committee kind of left you a handbook, guys. These are the things that we were unable to include in the last one, and this is some things you guys need to look at. And then did you put a little handbook together, some breadcrumbs for the next committee?
Well, I'll go first on that one because I'm in my office and it's kind of messy because that's my comfort zone, I guess. I didn't know I was going to be on camera. I would have cleaned it up a little bit. But anyway, I have this big box sitting to the left, this big whole box full of stuff that Chuck Schwab sent me. I mean, if you actually look through it, it's amazing the amount of work that was done by him and his colleagues that's embedded in there. So there really is a lot of that. I think in the chapters we tried to leave those breadcrumbs, cookies for people. So I'll let the others continue on that one.
I think in some cases it's large loaves of bread, not just crumbs that we left. One of the nice things, I guess, or one of the advances maybe that occurred more on a mechanical side or an approach side is that the modeling techniques and the approaches that were available and sort of had been worked out 20 years ago, they're nothing like today. I mean, there's been huge strides in that. Of course, there's been huge increases in computing power, and that's allowed people to develop these more robust algorithms. And so because we spent that time and effort to collect the data, we had the ability to cross-check things. So another data set that got pulled in was Hélène with, I think, again, Roger and others had collected a data set on splanchnic metabolism. And so I think there was 120 treatment means or something in that, and so we had diets where they had fed the diet and then measured portal appearance. So we could take, after Jeff got through with sort of going through the RUP and the microbial digestibility, and then looking at whether the amino acid digestibility was different, we could then go and look and see whether the portal appearance actually balanced with that. And it did, which was the problem before that Jeff was referring to, is that when they got done with all this factorial stuff of the RUP and the protein and all the amino acids and everything, it didn't actually match the flow even in the gut. And so they had to do an empirical correction. So not only do we match the flow in the gut, when we got done, we also matched the portal appearance by and large as well. So we were able to cross-check most everything along the way to have much more faith now. And of course, that just took a lot more computing time and a lot more effort on everyone's part.
Jeff, you've touched on protein supply. If we could circle back on that just a little bit, what would you say are some of the 2 or 3 biggest changes or revelations related to protein supply that's in this NRC as opposed to the last one?
I think one of the big things is the way we compute the passage rate. That's very different. The previous one was done in a very robust way, so not to diminish that, but there really is kind of no actual way to measure the passage rate of the so-called potentially degradable fraction. So actually, I'll have to give kudos to Mark on that. But what we did then was make sure that the non-ammonia, non-microbial nitrogen fraction, that's our best estimate of what would be in bypass protein. Then we corrected that for endogenous protein with equations that Hélène had derived. So this corrected version, Mark was able to then derive passage rates that on the whole would be able to resolve, and therefore we'd get RUPs that would match up with the corrected non-ammonia, non-microbial nitrogen fraction. That's what helps so it's sort of like two trains, one from the east, one from the west, and they meet somewhere, when they're laying the track. That really helped that to work. So to me, that was a big advantage. I wish we had a better way to do that mechanistically, but it's just the state of the way it is. We also tried to make the microbial protein equation more mechanistic. The previous one, well, it's not just that one, but also Beef NRC and others have done this. They've tried to match up energy in the total tract, looking at total digestible nutrients, and relate that as the main source of energy for microbial protein produced in the rumen. Statistically, it works out pretty well. But, of course, mechanistically, you can't have digested carbohydrate in the small intestine support microbial protein earlier. So there's some things like that I think were definite improvements, at least to let, you talked before about the cookies. I think to leave the groundwork for people to improve that. Like one place that we weren't able to do as well as I would like is to account for starch digestibility as being affected by how we process grains. We all know that affects things, but we just weren't able to make that resolve in a way that that connected all the pieces together. So somebody in the future hopefully will be able to do that and lay it in the groundwork of a more mechanistic microbial protein equation. That's my hope. The amino acid profile is different. Again, working with Hélène and a student of hers, we're accounting for protozoal flow. That's important, especially for lysine, because protozoa have a lot more lysine than bacteria. So we're now attributing microbial protein source as a little bit better source of lysine. So that, of course, affects what you have to compensate for in the RUP fraction. So there's some real nice gains there. I'm sure there are gaps that people will help to fill in later. That's kind of where I'm at on that. Other guys, you want to add to that?
I think on the post-absorptive side, the milk protein equation was a big challenge, really, and that occupied a lot of our time, and then because of the way we sort of approached it, Hélène came up with a way to address, okay, well, how do we back calculate what MP is, or what the flow of it is, and even today, I think it's better to think of it as more of a net protein system than it is an MP system, because to try to get to the middle, you're coming from both ends, right? And the middle is the least reliable part, probably, but I think the whole system, is consistent all the way through. And so whether we have exactly the right requirement and exactly the right MP supply, I don't know. But I definitely know that feed to milk is overall correct. Okay.
Yeah, that's a good point. One of the points I made at the Discover conference is, we really never do measure digestibility of RUP. We predict it in all the data, even the flow studies that we picked through to get these. Almost nobody sticks in an ileal cannula and derives that. So we had to predict it. The data are more robust, and if they're off a little bit, then they're off in a way that's self-correcting by the time you get to milk protein in the way that Mark described. I really feel like that's an advantage, and the committee was always really careful to try to account for that. So anyway, wherever it's wrong, hopefully it's wrong in enough of a way that's sort of self-correcting, and we get good prediction results.
Did you guys talk any about the next NRC and what the process had looked like? This last one took 7 years, right? And it was, should we start now? Should we start that process now?
The original goal was to have them every 10 years. So yeah, you're about a year away from getting started, because it takes a year to raise the money and get the committee selected. It takes another year to get going. So yeah, I think you need to start next year.
Yeah.
Different people, though.
Yeah, I was gonna say Jeff said he'd chair the next one.
I was just saying that's one point too, that we didn't really have time with the pandemic and everything, just to have like a wrap-up meeting with the whole committee that we're supposed to have once everything will be published and we'll have the software organized and everything that we're still working on. It will be to have like a post-publication meeting where we're gonna put all those thoughts together, and as you said, how should we make it happen for the next time. But what we really tried to do in this version is to have like I would call really a biology-based model on which it's going to be easier to add things or to change things or to move things compared to what it was before. So that's one thing that Mark really worked hard on, just to make sure that it's going to be much more easier actually for the next committee to take what we've been doing and just move things along as knowledge will be gained, as we know that it will happen. So I think that hopefully with that basis, it's going to be easier to make additions, deletions, or whatever than it was before. So, but as you said, this is really a big challenge, is to think of how that should be done the next time, so it doesn't take as long, and that it's, as Mark was saying, it was really a much larger commitment for everybody involved than we all thought it was to be when we agreed on. So we'll see how it goes. But we really need this wrap-up meeting actually, just to give a hand to not to give a hand, but to help the next committee.
Is the ingredient database, is that an ongoing project, or do you start from scratch with that as well?
It's sort of an ongoing project. Basically, National Animal Nutrition Program does a lot of that work for us. Well, at least the start of it. Okay, they gather up commercial data and they create an average ingredient. Of course, then there's a lot of fill-in stuff, like you're not getting getting KDs and KPs from that database. So that's stuff that Jeff had to work on, and amino acids don't come from that database, at least today. And so that was stuff that we had to get from elsewhere. So it is ongoing, and they're collecting it from commercial labs. So I think the proximate nutrients are much more reflective now than what they used to be.
And one thing that as it becomes more data driven, as we get more and more new data that will be added, they're also reflecting more of the nutrient composition of the feeds that are being used today. So what comes to mind, for example, is the corn silage that was fed 30 years ago isn't the same as the corn silage fed today. There's more starch and so on. And so it needs to be ongoing to help to continue to, sort of converge to be closer to what we're actually doing. So a lot of improvements in that way, like Mark said, with the feed testing.
Well, I think it should eventually, for the next committee, it may allow them to consider some regional differences too, because I'm pretty sure, Jeff, the corn silage that we grow in the South, with our weather conditions, is not the same corn silage you grow in the upper Midwest either. And we never get as good a production out of it as you guys do.
Yeah, you guys talk about the changes in feedstuffs. Our company, Balchem, recently did a survey where we analyzed methyl donors in, I think it was corn, soybean, I don't remember what else, clay, and we found out that it was significantly lower than the currently in the NRC. So, I mean, I can imagine if it's true of methyl donors, it's got to be true of a lot of different nutrients, even some that we may not be routinely analyzing for.
Yeah, I think once you get off the proximate nutrients, then our level of confidence in what we have goes down considerably. Just to come back quickly to that, the overall process, I mean, industry needs to push for NASM to do another one, okay? I mean, if we think we really need to start in another 2 or 3 years, it has to be, I think, pushed by industry, okay? Because who else is going to do it, right? It requires some money. It requires a commitment. It requires somebody to ask them to do it. They aren't set up to do it automatically. They're set up to respond to requests.
Yeah, good point.
Yeah, and during the Discover conference, the last session, there was discussion around this. I thought that was a really good discussion at the end of the conference.
But if everybody sits around and waits for the next one to happen, it's not going to happen. Okay, somebody has to actually drive the process forward to get it started.
Right.
You know, the nutrient requirements are supposed to be established on data. You know, you don't get to just say, I think it should be about this. You have to do the best you can to find those data. And we're not supposed to add safety factors. We're not supposed to be doing fudging. And that means that we need to have improvements where they're needed. So I couldn't agree more with what Mark said. It needs to be derived from industry for industry.
Jeff, as far as the protein supply piece again, how's non-protein nitrogen factored into the equation?
It's basically, if it's all soluble protein, there's a certain amount of that flows to the intestine. And I maybe Mark wants to address it, or Hélène, but I don't think we really address it as much as maybe we could. But at least this year, we allow some of the so-called A fraction, the soluble protein, to pass to the intestine. And Mark derived that equation, and maybe he's better to talk about this question.
Yeah, I mean, I think that, again, was more of a concerted effort, because one of Jeff's colleagues at Ohio State worked on that as well for a little bit. But essentially, I mean, there's several pieces of information in the literature that suggest that some of this A fraction should pass, right? It can't all be degraded. And even if you take like 1,000% per hour degradation rate, and you pair that with like a 15% liquid passage rate, 10% should pass, okay? And statistically, that's what we ended up deriving, was 10% without any guidance, right? That's what it comes back as. So I think that helped a little bit. You know, the KPs were too high. They should, you know, again, I think Jeff was alluding to this. When we measure KP most of the time, and certainly what the old NRC was based on, was the KP for marked particle passage. Well, that doesn't mean that's what the protein passage rate is, okay? That's what those marked particles are. And of course, those are heavily influenced by fiber and, you know, other nutrients. So we ended up with a different KP than what we had before. And it, you know, it essentially reduces the RUP content of all of the feeds pretty much, although, you know, more for the concentrates than it did from the forages, correct, Jeff?
Yeah, they're both decreased. So, you know, I don't exactly remember which was more, but yeah, both.
We basically just, that one actually didn't require much discussion. We just did it, then went down the road, right? And then once we had that, it allowed us to get, you know, our KDs and KPs. So.
Jeff, before we leave the protein supply section, so all the changes that were made from one version to the next, how will that manifest itself in the field? What will nutritionists, dairy farmers see that's different from what we were doing before, based on your recommendations?
Well, one of the things I think, you know, we grounded all the flow data into you know, we used all actual flow data to do that. And one of the things that really strikes me is, you know, I've heard it said before, the best source of protein for a cow is starch. But as you add more starch, you tend to, you know, actually lower the efficiency of microbial protein synthesis. So you don't get according to the prediction, you don't get as much as you might think. So one of the things I think we're going to see is we really need to have a good balance between rumen-degraded starch and you know, effective fiber and forage. So I think it's going to emphasize the role of forage, not just to keep the rumen healthy, but also as a source of microbial protein. So that's one of the things I anticipate will work out. There's some issues with digestibility of the RUP. We have to use book values that we had. Like I said, we don't have a bunch of data from studies for that. So I think on that standpoint, people are still going to have to be testing for it to make sure that the sources they're using are appropriate and, you know, highly digestible at the RUP fraction. So I think we're going to continue to see testing for those kind of things. And I guess that's the best way I can answer it. Maybe if one of the other two wants to chime in and help me out, but that's how I would think of it.
Jeff, I'm curious. What methods do you recommend for testing RUP digestibility?
That's a tough one. So we're still trying to figure out the best way to do this for all feeds. So I kind of like the mobile bag technique as a way to, for some feeds that there's really not a good way to do otherwise. So we're going to be doing some of that, but that's really hard to make work in the field. So we're still stuck kind of with the same techniques. There's the so-called 3-step or the kind like that. The Cornell folks have theirs that's pretty similar to that and pretty well worked out. So I think we're going to still see the same kind of approaches. I don't see a lot of changes in what we're doing, just that we need to continue to keep doing it.
Hélène, if we can kind of maybe start talking a little bit about the protein and amino acid requirements. Understand that you were fundamental in writing that section of the chapter. Can you talk a little bit about some of the challenges that you had with that?
Actually, I'm just going to go one step back. If we look at the outputs of net protein that were predicted by previous models, like metabolic fecal output or urinary endogenous proteins, actually, all that was based on a very good paper from Swanson, but that dated from 1977. So we thought that this knowledge actually there was enough knowledge actually to update those predictions. And also, I think we have a better assessment of what actually is metabolic fecal protein and what should be endogenous urinary protein. So we really started from the biology to revisit those numbers. And they were really changed a lot. Like endogenous urinary output is twice as much as it was previously, whereas metabolic fecal output is much lower. And also, again, based on biology, we decided to assign to each of the protein which is being exported out of the animal, which is creating a demand on net protein or on amino acid, and efficiency that would be similar across the functions, again, based on biology of how amino acids are being handled within the animal. So that meant that we needed not only to quantify the net protein that were secreted or gained, but also what was their amino acid composition. So we spent a fair bit of time on that, and we also assigned to those amino acid composition that we obtained from protein hydrolysis, as Jeff was saying before, a recovery factor to account for incomplete recovery. We all know that it's not new, but we kind of forget all things that when you run an hydrolysis for 24 hours, you get numbers for all of the amino acid, but actually it's more of a compromise. It's not good. It's not perfect for an amino acid. If you want to rank feed ingredients, that's going to be good. But if you really want to make a cycle analysis and see how much is being digested, how much goes into milk, how much is being absorbed, then you have really to take into account that your protein hydrolysis actually does not yield the total amount of each amino acid which is into the protein. So we also added that factor into the amino acid composition. And then by a factorial approach, we could estimate what was the net output or the net demand of each amino acid. And then based on the database that we had and on some studies, there isn't that many looking at splanchnic metabolism, what was the efficiency of utilization of each amino acid, because there is a large variation which depends not only on the amino acid supply, but also on the energy supply, which is something that was not considered in the previous model where we had a fixed efficiency. So I would have liked to pursue a little bit more the issue with the efficiency of use, but we needed to put a dot at some time point. So we'll publish papers later on, but at least we came with a target efficiency that we proposed for what we call an adequate energy supply that would complement the prediction of milk protein yield that Mark developed, just to have a guideline of what should be the efficiency for each amino acid that we should target. And if you calculate the efficiency based on the prediction of the supply and the output of milk that you want, what would be the needed theoretical efficiency? And if it's much larger than the target efficiency, it's telling us that the supply might be limiting. But it's not just like one amino acid. We want to look at in a holistic manner as a whole, including the energy supply as well.
We had a webinar with Dr. Chris Reynolds from Reading University, and there was a question asked, Mark, related to your comments about that there's not really, the whole barrel and stave concept is no longer valid. And the question was, and Clay, I don't remember exactly what it was, the notion of individual amino acid requirements now passé.
Single limiting amino acid.
I mean, all the mechanistic data, that's been collected at the cellular level and even some at the tissue level says, yeah, that's not how it works. So it's certainly an easy way to describe that, process, because it is sort of a complicated one. So, I wish I could come up with some analogy that would go into every textbook in the world for the next 50 or 80 years as well. Okay. So, and not only for animals, but it's for plants and everyone. I mean, everybody used that. In fact, it derived from plants originally. So I think if you think about it from an evolutionary standpoint, if that cow was, or any mammalian organism, was that sensitive to any single amino acid, they would have all fallen over every time there was a little shortage of food supply and died, right? And they would have died out as a species. So they had to evolve methods to mitigate a deficiency, or what we think was a deficiency, right? So if the cow's eating a diet and it's short of methionine, it's going to say, well, I'm going to conserve methionine. I will shut down use of methionine as much as possible at the splanchnic tissues, and I'll shut it down other places, and I'll ramp up my affinity for methionine at the mammary, trying to protect my investment in this calf, right? And so you have all these mechanisms that always resist. If you put too much methionine in, it does the opposite. It increases its catabolism. It resists taking it up at the mammary. I mean, it does still take up more, and you push some more milk out, but it's not as steep a slopes in general as what you would have envisioned with the old sort of very fragile system, I guess you might call it. So I think that concept, at the very least, has to be changed. Lou chastised me after the meeting, in Chicago, and said, if you really want something new to be taken up by people, you can't base it on the old concept. You just have to get rid of the old concept and blow it up and then build a new one. So I was trying to morph it into a leaky barrel, but he doesn't like that. So I don't know.
Okay, I thought it was a good idea.
I like the leaky barrel.
I think it's where the concept of efficiency is interesting too, because one amino acid is not limiting per se, but the cow cannot put out more than it digests or that it absorbs unless it's taking some on its body reserves. So there is always still a physiological limit that we have to acknowledge. And I think this is where the calculation of the efficiency is making some sense. As if you're having an efficiency of 95%, that if the cow is achieving that, it's going to be taking some of the amino acid on its muscle, or it will not get there. So, but between 75 or 70 or 80%, depending of the amino acid, there's a margin where the cow can adjust actually. And I think this is really where in the future we will have more studies, and we'll have more studies, because in one of the questions you asked first what type of study we were missing, and this is one of the things that we were really craving of, were those type of studies on amino acid where we would have only one amino acid moving at different doses, because often we have one level here, one level there. So, but what happens between the two levels, we don't have a clue, or we have all of the amino acid moving together. So the response that we observed, we have to pull some hypotheses and to derive some equations. But there was a very limited number of studies where actually we had enough data that we're moving one amino acid supply at different doses. Is really something that we do miss. And also I think we all needed to start somewhere. So first, people were looking at like a silo of protein and amino acid and a silo of energy. But now I think we're really trying to merge these two because, we'll see it with the equations with Mark, and also in the estimation of the efficiency of utilization of MP or amino acid. Energy is really a key factor in the efficiency of utilization of the amino acid as well. So I think that we need to merge the two now, and also we get into the type of energy, and so a
Can
lot
I,
of work for new scientists.
Scott, can I just add a comment?
Yeah, absolutely.
I think, I really have to give kudos to my two colleagues. They really improved the efficiency concept and the quantification of how to use it. That was, listeners need to remember that was a fixed value in the previous NRC. Everyone knew that it shouldn't be fixed. If you feed more protein, then the efficiency of transferring that into milk goes down, and vice versa if you feed less. So that's really a big improvement. And the other thing, whatever model he winds up to show it, the leaky barrel or whatever, people need to remember that not all of the amino acid goes into a protein that needs that amino acid at that place. There's a whole bunch of amino acid that does a bunch of other things. Like you talked about the C1 transfers. I just read yesterday from my amino acid metabolism class that something like only 20% of methionine is actually used in protein synthesis. So these other processes get prioritized as you shift, as you like what Mark was talking about, whether it's maintenance of the calf or maintenance of genetics for milk protein yield. So again, my kudos to my colleagues. That's really, I'm not sure people are going to see it, but that's really there. And it's also the foundation of what Chris Reynolds and his colleagues are trying to do to lower protein. And that is how do you maximize efficiency and not have some kind of issue backfire through that. So anyway, I just had to say that. So
No, thank you for that.
So Mark, can you maybe, for our listeners here, can you remind us what is the what's the new milk protein yield equation in NASEM?
It's just a simple multiple linear regression equation, okay? With like 13 terms in it and a quadratic term, but it, basically it treats each of the nutrients as separate contributors to the overall, right? So if you think about protein synthesis, it's like an assembly line, right? We can't assemble. Well, right now we most of us know if you're trying to buy a car, you can't assemble a working car if you don't have enough chips to go in the car. Okay, it doesn't matter how many wheels you have, how many doors you have, how many seats you have, the car still will not go. Okay, and so I think that's the same kind of idea here, is that assembly line can sort of stutter maybe. And I'm not saying that this is actually what happens, but you can think about it that way, that it will stutter every time it gets to one of those nutrients that it needs that is in shorter supply, and it'll slow the overall line down. And particularly if you start thinking about it as thousands and thousands of lines that are operating in that tissue, you get some of them slowing down. Okay, and you provide a little more nutrient and it goes up. So the equation has to reflect that. It has to have this independent and additive stuff. And then there's some quadratic stuff that is interactive, but by and large, that was the best equation. Okay, it wasn't magnificently better than some of the other approaches that may be considered more older concepts, but it did come out statistically the best approach, and it fits all of our conceptual data.
So from a nutrient standpoint, I'll paraphrase here. So milk protein yield, it's an energy driven process, and the key amino acids, methionine, lysine, histidine, isoleucine, and leucine, is that correct?
Yeah. Did we miss anyone, Hélène? We had 5 of them, I think, that ended up in the equation. It's not to say the other ones are not important, it's just the data were not adequate to pull them out uniquely. So we had isoleucine, leucine, histidine, lysine, methionine. Yeah, those are the 5. Okay.
So one question that we've been getting from people is there a weighting to those amino acids as far as milk protein yield?
Yeah, each of those terms in the model has its own coefficient. So the ones that you can expect to see bigger responses per unit of absorbed supply are methionine and histidine. They both generate about a 2-to-1 response, right? 2 grams of protein per gram of absorbed. Isoleucine and leucine and lysine are all closer to 1. So if you're going to add something, if it's expensive, then pick the one that has the biggest response, right? If lysine's cheap or something else is cheap, I know like there's been interest in tryptophan and threonine, or at least tryptophan, if I remember right, because it's really cheap to produce, I guess. It's a byproduct of some other things they're already doing. So they'd love to have tryptophan in it. Well, there's no, to Jeff's point, there's no data like, I think the feed data maybe is okay on tryptophan. There is not a shred of believable tryptophan data from mouth to milk, okay, after that. I mean, maybe milk's okay and mouth's okay, but nothing in between. There was nothing cross-check on that all the way through. Yeah. It showed up as significant in the milk protein equation, but none of us trust it, okay? So it could just be an artifact of something else.
Yeah, and I would just add, this is my bias, obviously, check for what is the least expensive, but then just make sure that you check the efficiency, because if you put a lot of lysine, for example, because it's really cheap, but then you don't have enough methionine, you can end up with a nice predicted milk protein yield, but your efficiency of methionine is at 95% because you put all cheap stuff that has a lot of lysine but not enough methionine. Then you're going to be away from the target efficiency. So it really needs to be balanced with the two concepts.
Yeah, and that's something that we struggled with and that we didn't really have adequate data to address, is that sort of interaction, right? I mean, the stats say there's not really a very strong interaction, but we know there is. When you get really far away, I mean, if you create an imbalance, it's going to have negative effects on the animal. Maybe that's only at intake, I don't know, but we know you can't just like say, okay, well, let's put in, a kilogram of methionine, okay? Even if we could afford it, it's not going to make the animal operate well. So yeah. We struggled with how do we sort of come up with this idea that there's a range, right? We don't have to be right on this specific number, but there is a range of the ratios that probably need to occur amongst the amino acids. And I think, Helene's idea of using these target efficiencies helps you keep on that, sort of on that ridge, maybe you might call it. You got a ridge that's wandering around, and so you have a ridge there that's these target efficiencies. We can drop off of that ridge and still be fine, but we can't drop off too far, okay? And that's the part we couldn't pull out of the data. The data just were not strong enough to tell us how far away, because everyone fed according to the old NRC in most cases, right? So we don't have a lot of data off the top of that ridge. We got lots of data along that ridge, but not off the top of it.
Yeah, and also for those amino acids that were not included, for example, they didn't come out being significant. I think a lot of that is due to the fact that a large part of the database, actually, where we had one amino acid that was increased, the supply was increased, often it was like a change in NP supply, so that was all of the amino acids that were also increased. We didn't have that much data, for example, on phenylalanine. And when you look at the data, for example, maybe like the phenylalanine content of the microbial protein is so high, actually, that most of the times we do have plenty of phenylalanine. But we know that if we run a deletion study of phenylalanine, we might decrease milk protein yield. But in theory, it is very unlikely that this will happen with a normal dairy ration. So we couldn't catch that with a global equation. It didn't come out to be significant. But if you run 2 or 3 studies where you delete phenylalanine, it's going to be crucial, and it's going to be really important to maintain your milk production. But generally, in the whole data set that we have, because of this limited number of studies that we have, with just one amino acid changing, we could never catch that statistically.
So even though phenylalanine is not in the equation as a driver, that doesn't mean you can ignore it, I think is what you're saying, okay? It still needs to be along that ridge someplace, okay?
Mm-hmm. Jeff, I want to go back to a comment you made a few minutes ago about how only 20% of methionine is used for milk protein synthesis. So if a cow is deficient in methionine, what's the priority for use for methionine? What's it being used for metabolically?
Well, I was reading it was actually for humans, so it's a bunch of human nutrition people. So I don't want to transfer that to milk protein. But I was just making the point, like it's a bunch of it's used for cysteine or other functions, and that all those functions can be reprioritized as they need to be. So otherwise, I think probably it'd be better. Maybe Mark, you want to try to answer that question on the efficiency?
Yeah, I mean, I think that it seems like the more we go down this road, that there's not this, we would like to say, okay, well, first we're going to do this, and then second we're going to do that. I don't think the cow or any of the organisms work that way. It says, I got to do all these things at the same time, and I'm going to manipulate and slide things one way or the other a little bit, but not turn off in general. I mean, unless things get really bad, right? I mean, if you restrict nutrients enough, she will turn off milk production, okay? And same way with gestation, it'll abort if you make the deficiency enough. So I think it's, in terms of the methionine thing, I think there's a certain amount that has to be used for that methyl donor, and it probably is going to depend on other things as well. And I really don't know, but there's a lot of these nutrients that we don't know that much about, and we certainly don't know the interactions. Okay, even with the amino acids, we're still trying to figure out the primary drivers, and we just don't have enough data to figure out those interactions. We can't keep doing. I know we're trying to stay a little bit more applied here, but we can't keep doing 2 by 2 factorials forever and think that we're going to work out very many of these interactions on a surface, right? We're going to get 2 points, but to try to pull that out and make a surface response to answer your question, really hard to do.
I guess maybe one final question is, obviously, we have herds out there averaging well in excess of 100 pounds of milk a day, and certainly lots of cows and even groups on dairies that are 150 pounds or more. I'm curious, how robust is the database, from a requirement standpoint for these high-end producers?
We had, I think the upper end of the milk, Helen, if I remember right, is like 110 pounds or something like that, 115. We don't have a ton of data up there, but we have data up there. But, if you have a herd that's averaging 100, then you got pens that are averaging 120, right? Which means
Right.
Individuals are at 150 or something like that. We don't have any data like that. I mean, there might have been in those treatment means, when they were put together, but we can't recreate that out of treatment means from the literature. So I think we have better mechanisms captured now, which would give one a little more confidence that it'll operate okay, somewhat outside of its range. But I think it goes back to buyer beware. Okay, we define what the range is. Okay, you have to decide how much faith you're going to put in it when you start operating very far outside of that range.
But I would just add that in terms of efficiency, actually, even if the cow is producing more, actually, it's going to eat more. And I think that in terms of efficiency, we should be pretty much on the same type of global organization. And also, Mark, you developed like a scaling factor to predict those milk protein yield for the cows. Actually, as we were discussing, we had this discussion a few times, we're working with cows of the past with published data for cows of the future. And obviously, the cows that have given us their data are producing less than the cows that we want to feed now. And Mark, you developed a factor actually to take into account that the prediction actually needs to include a factor that is taking into account the rolling herd average of the herd.
I guess we're kind of getting to the end here. Is there anything that we haven't covered related to the protein and amino acid chapter that you think the audience needs to know about?
I think just to reiterate that, both at the microbial level and at the milk protein level, and probably at growth and the other functions as well, there's this interaction, okay, between energy and amino acids. we only have an energy and protein at the microbes, but that doesn't rule out the fact that there might be amino acids that are, that those microbes will respond to that we just can't see. Okay, but I think we have to stop thinking about this first limiting nutrient. I know you asked me that question before, and I sort of, got off into the barrel stuff. But if we continue to think that way from an application standpoint, you're going to be frustrated with this model, because you'll be able to change the energy and think that, okay, well, I'm going to have this big change, and it will be held up by the other nutrients that are there. So it requires a little bit different thought process.
Very well. Well, with that, we'll call last call. I'm going to ask each of you to give us kind of one or two real key takeaway applied messages from the chapter that a nutritionist or dairy farmer can use right away. And Clay, why don't we start with you?
So yeah, I mean, I'm certainly I'm excited about the approach here to milk protein yield and being able to apply that in the field with the new equation that's there. I do want to ask a question to the group, though, maybe for my last point here. I am curious, and Scott alluded to this a little bit earlier, but I'm curious how you think this will be applied in the field as far as software. Do you see the NRC, the NASEM software being applied, or is there a plan in place to basically get this into some of the other models that are some of the other software programs that are out there?
Well, I know from when we were looking for programming support to do the software, there was a couple of the companies, the commercial vendors, that did express interest in providing that support, and at least one of them was going to charge less money provided they could get a license to put it into their software. Okay? So I think that is the route, because a chunk of the money does come from industry. Of course, those vendors do not want NASEM acting as a competitor in the marketplace, nor does NASEM want to do that. Okay, they don't have software support. In fact, if something breaks on that software, National Animal Nutrition Program has to fix it most of the time. Okay, because they don't have it. It's like an end product. They wouldn't even be bothered if there was no software. It's us that say we have to have software to teach and do extension. So that software is a teaching and extension tool. It is not a commercial application tool that you're going to work with more than just a herd or two. So I hope it gets put into these other programs and done correctly.
Hélène, what kind of words of wisdom do you have for our audience?
Well, I think that we need to learn how to use the efficiency of utilization, how to calculate it, how to deal with it, and better learn how it's being affected by the energy as well as the amino acid supply. I think we gave some target efficiencies, but as for each amino acid, but as Mark was saying, it's likely not just like a very tiny number. We don't have like the uncertainty around it. There is some uncertainty around it, but I think that we really need to make sure that we're not trying to make the cow make more use of the amino acid that she has absorbed than what she can. So I think that this is like the type of guideline that we need to keep in mind, and we're happy that we could provide it for all the essential amino acids in a factorial manner. So I think that's interesting, but because if we find, for example, afterwards that we have a better quantification of methionine, for example, as methylone or whatever, we can just add it in terms of on the model, and we can have another efficiency or another requirement and work with that.
Thank you. Jeff, how about you?
Well, just thinking and listening to what the other two said, I think it will be used and compared. One of the strengths, I think, will be when people go to run simulations, they'll see what this model predicts compared with another one, and then they'll try those under different situations, and they'll figure that out. I'm always mindful of what somebody who I know who fed a lot of cows, he goes, I really like the models because they help me think. And so I think there's going to be a certain amount of that people are going to get used to, and they're going to continue to try. The other thing I wanted to point out is we have a charge to use data, but we also are scientists and we also want it to work. So we tried to find the best way that we could to make all those things function. And so one of the key points that I think is going to lead into the future, maybe is more indirect answering your question. There's going to be study after study after study that does comparisons, and study after study that now is designing new objectives based on what this model says. And I think that's a big gain that's eventually going to be helping people into the future. So at least that's my hope.
Thank you. Dr. Hanigan, final words.
I'm glad it's done. My wife thinks it will never get done. She doesn't think we're done yet, nor will we ever get done. But, I certainly want to, give a lot of credit to Bill and Weiss and Rich Erdman as well. I mean, particularly Bill, on the software here and all he pretty much did a lot, in my opinion, did almost all the editing on the whole book. And it's, I think, 525 pages or something like that. And he just read through this modeling chapter again, this week. And so I have trouble reading through that. Okay. I just don't want to look at it again. He's read through it several times. So really got to give a lot of credit to him. But from a field application standpoint, I think that, the old equations were too sensitive. They weren't correct on average, and they also were too sensitive. So it told you if you were short on protein, you were going to have this massive loss in milk production, and it didn't happen. Okay, and it also told you're going to get this massive gain if you fix the problem, and that didn't happen either. Okay, so I think probably the take-home message is that probably explore a bit more outside of the bounds. I'm not saying you should do stupid things, but the cows are pretty robust. So if you have a situation that calls for deviating a little bit from what the model says you should do, I would give it a try. Just make sure you position things with the producer correctly, that, hey, we're going to try this. This might happen, but it's not going to be as bad as what you would have thought.
Great input. Folks, I want to thank you for joining us here this afternoon at the Real Science Exchange. It's been enjoyable. It's been a long time coming, but it's been worth the wait. Did not disappoint. Also want to thank our loyal listeners for stopping by once again here at the Exchange. Hopefully you heard something new, something interesting, something you can take back to your business. Also, as a reminder, we will continue to break down the new 2021 edition of the NRC over the next coming weeks. Be sure to subscribe so you don't miss any of the new episodes. If you'd like to pre-order a copy of the new Dairy NRC and receive a 25% discount, visit balchem.com/realscience and click on the NRC series for a link and the discount code. If you like what you heard, please remember to hit the 5-star rating on your way out. Don't forget to request your Real Science Exchange t-shirt. You just need to like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Our Real Science lecture series of webinars continue with the ruminant-focused topics on the first Tuesday of every month. Visit balchem.com/realscience to see the upcoming events and past topics. We hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.